Our next speaker is Hunter Everett from North Carolina State University.
All right, thank you, Dr. Zitnitsky. Like she said, my name is Hunter Everett. I'm a third-year veterinary student at North Carolina State University, and I'm here to share my findings on the impact of Lawsonia intracellularis maternally derived antibodies on the serological response post-vaccination. So Lawsonia intracellularis, or ileitis as you'll also hear me call it, is an obligate intracellular rod-shaped gram-negative bacteria. It was discovered back in 1993 and can only be grown in cell cultures. So what makes Lawsonia so important is that Winkleman and others have reported that up to 96% of farms in the United States could be infected with ileitis. And so if a pig's infected after about 10 weeks of age, what we would see is that 20 to 40% of those pigs would show clinical signs and the other 60 to 80% would be subclinical. And so that subclinical manifestation is going to present with a 39% lower average daily gain and a 27% lower feed conversion ratio. And so what that means economically for our farmers is that they could lose up to $5 per infected pig due to a decreased carcass weight and feed efficiency and an increased morbidity and mortality. And so what really got me interested in ileitis and more so the effect that vaccination can have is that in current literature, there's a lack of knowledge on the effect of maternal antibodies, mainly on vaccination, and that we found in our last study that maternal antibodies for ileitis can last anywhere from 6 to 12 weeks, making them a prime candidate for interactions. And so the objective of my study was to determine the post-vaccination serological response of different vaccination or injectable ileitis vaccination programs in the face of Lawsonia maternal antibodies. So the product that I used was the Porcellus product. It's labeled for two main uses, the first being a single dose 2 ml injection that's given after 3 weeks of age, and the second being a 2-dose program where the first meal is given after 3 days of age and the second meal is given 3 weeks later. And so the Porcellus product is the first IM ileitis vaccine, and it's a bacterium vaccine with a 20-week duration of immunity. So the flow that these pigs came from was selected based on a previous history of diagnosed ileitis. Our pigs were farrowed out on a 4,800 head sow farm and then followed through the nursery and into the finishing house up until closeout. So our pigs on the sow farm were housed across 5 different rooms together by litter, and we ensured that no cross-fostering or fallback litters were allowed in order to maintain consistency between our treatment groups. So when we went in, we ended up enrolling 193 sows at the beginning of our study. Half of those sows were vaccinated 3 weeks pre-farrow with the Porcellus vaccine, and the other half were left as unvaccinated controls. And so all of those sows were bled at the time of vaccination and then again 2 weeks later to determine their antibody levels. And then so after everyone had farrowed, we ended up enrolling 100 final litters, with 50 of those litters coming from vaccinated dams and the other 50 coming from non-vaccinated controls. And from each litter, we took 3 gilts for our 3 different treatment groups. So the first treatment group was given a full dose 2 cc Porcellus vaccine at 3 weeks of age. The second group was given a split dose vaccine where we gave the first cc at 3 weeks and then the second cc at 7 weeks in the nursery. And then the final group was just given 1 cc of saline at 3 weeks of age. And so every 4 weeks, I would go in and take serum samples and fecal swabs on all 300 of our gilts. The serum samples were tested by IPMA and ELISA for Lawsonia, and the fecal swabs were going to be tested by qPCR. And so every 2 weeks, I would also go in and take oral fluids and screen those for flu, the coronaviruses, and then PRRS and ileitis as well. And so statistically, what we did was we took the fecal samples at 3 weeks and at 22 weeks of age, and we tested those by qPCR. And so those and the serology data were analyzed using a one-way ANOVA, and the positivity rates for serology and fecal samples were tested by chi-squared. And so one thing that I wanted to talk about before we got into the numbers are the differences between an ELISA and an IPMA and why we chose each of those tests. So the ELISA is an enzyme-linked immunosorbent assay. It's a competitive inhibition ELISA for ileitis, and it's really good for giving us a yes-no answer when it comes to that. The IPMA, on the other hand, is an immunoperoxidase monolayer assay. And so the best way that I have to describe that is that it's really similar to an IFA, except instead of being fluorescent, it's just going to be brown. And the reason we chose that was because in the past, we've seen some issues where we would have an age response with our ELISA. And so in our breeding age animals and our really young animals, we would see some false responses with that. And so the IPMA was another way to just kind of back check that. And it's also really good, since it's a titrational test, to give us a quantitative analysis. And so here we have a graph, and just to orient everybody before we get started, this is a comparison of antibody response. And so on the left, we have our test type and the day at which it was taken. And then in the orange or the pink, we have our MDA negative groups. And then in the green, we have our MDA positive groups. And so it looks like my boxes may have shifted a little, so I apologize for that. Just listen to what I say, not what I show, I guess. So the first point I want to draw your attention to is the 7-week time point. And so what we saw, this is going to be 4 weeks after our first vaccination. What we saw is that all of our treatment groups for our MDA negative pigs did show a significant serological response after that first vaccination. Whereas in our MDA positive pigs, none of our treatment groups showed a significant response. But then, however, at 11 weeks of age, what we saw was that our split dose vaccine for our MDA positives did in fact show a significant response, and that response was pretty equivalent to the full dose vaccine in our MDA negative groups. And then we also saw a significant response with our split dose vaccine in our MDA negative pigs to a little bit higher. And so here we have a really similar graph, except this time it's going to be a comparison of antibody positivity rates. And so the main thing that I want to point out here is that with our MDA negative groups, both of the treatment groups ended up having full antibody levels out until 15 weeks of age. With our MDA positive groups, the full dose group only had 1 positive at 15 weeks and only 17 out of 21 positives with the split dose vaccine. And so this, I think, was kind of one of my favorite parts about this study, because it really made me question some things. And so here we have a comparison of our fecal PCR rates. And so it's set up pretty similar, except on the left this time we have positivity rate, PCR values, and CT values. And so what we saw was that with both our MDA negative and our MDA positive groups, as long as our pigs got a vaccine, they had a pretty similar response, and they all decreased positivity rate, PCR value, and CT value. And so what that really does is kind of begs the question on whether or not we can infer protectivity from that serological response post-vaccination. And so with that being in mind, I think the next steps that I would like to see done with this information is to perform a controlled Lawsonia challenge study where we have staggered MDA and antibody levels in order to determine the true impact that we can get from serological response and protectivity. And so just to recap what we've learned and what I've shared, the split dose vaccination of MDA negative pigs will produce a more robust serological response than a single dose vaccination under these conditions. And then when vaccinating your MDA positive pigs with a single dose, we did not produce a serological response. But when a split dose vaccination of MDA positive pigs was given, we produced a response that was similar to a single dose vaccine of MDA negative pigs. And then I think the most important thing to keep in mind, based off this information, is that vaccination of both MDA positive and negative pigs reduced fecal shedding levels when compared to their controls. And so with that, I would like to first off thank Dr. Connie Gebhardt at the University of Minnesota for all of her help. We couldn't have done it without her analyzing all of our IPMAs. And I'd also like to thank Merck and Smithfield for all of their sponsorship and support during this project as well. And with that, I would like to thank you for your time and take any questions.
Thank you, Hunter. Any questions for Hunter?
Excuse me if I missed this, but the sows that you enrolled, how did you stratify on parity, or were they all the same parity?
Yeah, so the question was on kind of how we blocked or stratified our sows. And so, yeah, all of our sows were blocked as either gilts, P1s and 2s, or P3 pluses. And so for each block, had a similar, so we would have like an MDA negative gilt and an MDA positive gilt, and all of her pigs would be included in that block. And then her pigs would be housed together throughout the study.
Thanks.
Yep.
What was the question? I have one for you, Hunter. So then what would be your recommendation? Do we need to split those then? Is that what we are telling our clients?
Yeah, so if we're looking at the serological response, yes, we got the best response with a split dose vaccine, whether your pigs were MDA negative or MDA positive. But I think in the long run, right now, based on this data, we can't really say protectivity-wise whether or not that would make a difference. So I would say based on fecal data, as long as you're vaccinating your pigs, that's what I would ask for right now.
Thank you. Other questions? Thank you very much, Hunter.
Thank you.
And you get extra credit for saying my name right.
Wow.